Hey everybody, welcome back to General Biochemistry Lecture. This is part 2 of chapter 14. We've already covered glycolysis in the first part of this series. Now we're going to talk about the fates of pyruvate, gluconeogenesis, and how glycolysis and gluconeogenesis are regulated so that they're not fighting each other. We'll start with the fates of pyruvate. You'll remember that pyruvate is the end-all be-all. It's the end of glycolysis. We make 2 molecules of pyruvate. We make NADH along the way in glycolysis. That NADH must be recycled to regenerate NAD. So under aerobic conditions, pyruvate is oxidized to acetyl-CoA, and then you can do oxidative phosphorylation after that, and bam, right? So you get acetyl-CoA, citric acid cycle, oxidative phosphorylation. All things are great. If you're under anaerobic conditions or just really low oxygen, which is also called hypoxia, then pyruvate is reduced to lactate or ethanol. Fermentation is the general term for processes that extract energy, like ATP, but they don't actually consume oxygen or change the concentration of NAD or NADH. And the key here is change the concentration. So you may use NAD and make NADH, but then you may recycle it and regenerate NAD in the end. So that's totally fair in fermentation. Lactic acid fermentation is when pyruvate accepts electrons from NADH and it's reduced to lactate. That generates NAD, which is necessary for glycolysis. Ethanol fermentation or alcohol fermentation is when you take pyruvate and you further catabolize it to ethanol or some other alcohol. So let's talk about the Pasteur effect. The Pasteur effect is the effect where the rate and the total amount of glucose consumption under anaerobic conditions is much, much higher than under aerobic conditions. Now let's think about why this happens. Glycolysis, you make a couple of molecules of ATP, you make a couple of molecules of NADH. But that's not a whole lot of energy in the grand scheme of things when you compare the output of energy of glycolysis to the citric acid cycle and oxidative phosphorylation, which are both aerobic processes. We're talking about an order of magnitude greater of ATP in comparison to glycolysis. So if all you're relying on is anaerobic conditions and doing glycolysis for energy, you have to do a lot of glycolysis. You've got to burn through a ton of sugar to get the same kind of energy yield. The Warburg effect is the observation that tumor cells have a really high rate of glycolysis, and they actually use fermentation of glucose to make lactate even when there's oxygen around. And this is one of the things that's used to detect tumors and diagnose them. So you can look at the body and you can tell where you're using up a lot of sugar. So you can use a labeled sugar that will not actually get processed through glycolysis, but it will get taken up by cells that are rapidly undergoing glycolysis. And so you can take a scan of the body and look at the soft tissues. Then you can look at where the where that labeled sugar is in the body. And in the image that you have, that's number 2. So this one is the one that shows you where your labeled sugar is. All the dark spots is where the labeled sugar collects. And there are some places like the brain that solely rely on glycolysis for energy. In those places, you're going to see an accumulation already. But what you're generally looking for is places that normally don't use a lot of glucose relative to places like the brain. And then you can overlay these two to look at areas of increased glycolysis. And that's shown in kind of like the green is, kind of the lower end of the spectrum, progressing to yellow and red. So if you see red spots, then that means, okay, these places use a lot of glucose. There's a lot of that labeled sugar taken up, and you can start to pick apart where the cancer might be. You don't need to understand how to look at one of these scans because I am not a medical doctor at all. But you should understand the Warburg effect and why you can use this observation to diagnose tumors. So when we look at lactic acid fermentation, pyruvate accepts some electrons. Okay, and in this way, we can regenerate NAD, which is so critical if we're doing a lot of glycolysis. Lactate dehydrogenase catalyzes the reduction of pyruvate to lactate. So you'll notice that there's a ketone here. Using the dehydrogenase, you're going to regenerate NAD, so it's going to accept a hydride, and you're going to form lactate. Notice the delta G here, it's negative. So glycolysis, you're taking NAD and converting it to NADH. In order for it to become useful again in glycolysis, you need to regenerate that NAD. So that's where fermentation of pyruvate to lactate comes into play. And you can continually cycle that NAD and NADH so that there's no net change to the concentration of either of these cofactors. The other good thing about lactate is that it can be recycled. So lactate, once it's made, is carried through the body via the blood to the liver. And in the liver, you can actually take lactate and convert it into glucose. If you are an athlete, if you do a lot of sprinting and stuff like that, then you'll definitely already know like the burn. That's the lactic acid building up. During recovery, you're going to take that lactate and convert it to glucose so that you can replenish your glycogen stores and do some repair to your body. The release of all this lactic acid from the muscle into the blood causes acidification. So you can't really do that forever. That burn, again, is going to start to happen. And so if you're doing a really vigorous activity like sprinting, you can only do that but so long. So even if you're an Olympic athlete, you can only sprint at your very top speed for at best about a minute or so. Another form of fermentation is ethanol fermentation. So yeast and other microorganisms do this, and that's how they regenerate their NAD by reducing pyruvate to ethanol and then also making carbon dioxide. Now, if you like beer or bread, then you're familiar with this fermentation process, even if you didn't know it. So when you make bread, I love making bread. When you make bread and you use a little bit of yeast, you activate that yeast, maybe it's an instant dry yeast or something like that. It gets hydrated, the yeast wake up, shake themselves off, and they start to eat the sugars that are in the dough, and they start metabolizing them with glycolysis, just like us. But the difference is they make ethanol and carbon dioxide. That carbon dioxide, they kind of, I'll say burp out, but they're yeast. They don't actually burp. They just release gas. They do gas exchange. And that is what helps to lift the dough. So you're digesting the sugars and you're making ethanol and other intermediates from glycolysis. And all of these things, all the processes that the yeast does by taking in that sugar and digesting it, essentially, it gives all the bread different flavors. So you're tasting all the chemistry that the yeast are doing. And maybe that kind of ruins bread for you, but for me, it makes it like super cool. Bread is chemistry, and it's biochemistry. Anyway, so to do this, we need two things. We need pyruvate decarboxylase, and that is an enzyme that uses a new coenzyme that we have not talked about called TPP, but we're going to address it in a minute. And it also requires magnesium. So with this reaction, you're going to get rid of carbon dioxide. So that's a decarboxylase. Then you're going to use an alcohol dehydrogenase, which will replenish the NAD, and you make ethanol. The intermediate here is acetaldehyde, which is actually quite lethal if it builds up in your body. The overall equation for this, you're taking glucose, and this is taking into consideration glycolysis plus ethanol fermentation. Glucose, ADP, and inorganic phosphate. And we're making ethanol, carbon dioxide, and ATP. We don't really care about the water. So that's the overall equation that yeast and other microorganisms do. Now let's talk about some chemistry. Pyruvate decarboxylase. It catalyzes an irreversible reaction, the decarboxylation of pyruvate to acetaldehyde. You need magnesium, and you need a coenzyme called thiamine pyrophosphate. If you're looking at thiamine like, that looks familiar, you're right, it does. B vitamin. Then the alcohol dehydrogenase catalyzes the reduction of acetaldehyde to ethanol. So that means that ethanol, or the acetaldehyde, is going to accept a hydride from NADH, becoming ethanol, and then you've regenerated your NAD. The alcohol dehydrogenase we have shown here. It has a zinc in its active site, and that zinc helps to coordinate the oxygen in the ketone. And when I say ketone, I know that there's a hydrogen here, so the whole group would be an aldehyde. But if we're just looking at the carbonyl carbon and the oxygen, this is still a ketone. So when I say ketone, that's what I'm talking about. Okay. I'm going to erase those so that it doesn't get too cluttered. But just wanted to clarify. So this zinc helps to coordinate the substrate, and it also helps to polarize that oxygen to make the transfer of the hydride that much easier. So it makes this carbon even more ripe for attack. What happens is, within that nicotinamide ring, there's a nitrogen with a lone pair. And because of the resonance in this structure, you can have this lone pair saying, hey, you know what? I think I want to be part of a double bond. And so you rearrange the electrons such that you can kick out a hydride, which is a hydrogen with electrons, and attack this carbonyl carbon. The electrons in that double bond between the carbon and the oxygen go up to the oxygen. And again, that zinc helps to stabilize that. Then you add in another hydrogen ion from the atmosphere, from somewhere in the site. Remember, this is happening inside of a cell, and you've got water everywhere. There's all kinds of things that can donate a hydrogen. So a hydrogen gets donated, and you make the alcohol. We call it hydroxyl in biochem land. So this red hydrogen came from the NADH, and it was transferred as a hydride. The blue one came from water or something else in solution. Now let's talk about thiamine pyrophosphate, TPP. It's a coenzyme that's derived from vitamin B1. And the thiazolium ring plays a really big role in cleaving the bonds that are next to the carbonyl group. So the thiazolium ring is right here. And so that is the business end. All the other parts of TPP are pretty much used for specificity. It's more parts for the enzyme to hang on to and make interactions with to make sure that TPP doesn't go anywhere. Now let's talk about the mechanism. We've got TPP, and it can have resonance, all right? So you go from having this hydrogen here connected to the carbon, and there's a positive charge on the nitrogen. Then you can kick out a hydrogen and form a carbanion. So that's the carbanion. And it can attack the carbonyl carbon. Once it attacks the carbonyl carbon on pyruvate, so that's C2, then you have an intermediate that is attached to the TPP molecule. Then we can facilitate decarboxylation because you have resonance within this structure. So the electrons on the oxygen can go in here and form another double bond. And then these electrons can be relocalized to form a double bond between the carbon of the substrate and the carbon in the thiazolium ring. So once you make this intermediate, there is some resonance within this intermediate. That nitrogen in this position here that just accepted some electrons can now push those electrons back out and create that double bond between it and the carbon again, creating that imine. And then the electrons go up here to this carbon to create, again, a carbocation intermediate. When you have that carbocation structure, it can then take on a hydrogen and you're forming your product. You can then take another hydrogen off of this hydroxyl group. Those electrons go into forming a double bond between the carbon and the oxygen. And you have cleavage of your product from the thiazolium ring, from the TPP. And you release acetaldehyde. This is, I think, a really cool mechanism because you never really get to see carbon with a charge. So in this case, we've got a carbanion that does all the work. Know this mechanism. And so for right now, we're talking about glycolysis and the fates of pyruvate and all that in chapter 14. Thinking forward to the final, if you know a mechanism for how an enzyme that uses TPP works, I could give you a different enzyme that has a very similar mechanism, and you should be able to draw that. If you know what the substrate is and what the product is, you should know how to draw a decarboxylation reaction to release carbon dioxide and whatever the product is. So that's just a heads up for the future. For right now, just focus on understanding this process in terms of fermentation, ethanol fermentation. So this step is the one that makes the acetaldehyde, and then it goes to the dehydrogenase to make ethanol. So here are some examples of TPP-dependent reactions. So the synthesis of acetyl-CoA in the citric acid cycle, that requires decarboxylation, and you make acetyl-CoA. You can also make alpha-ketoglutarate within the citric acid cycle as well. Then there's also transketolase, and that is used in carbon assimilation. And that's in the pentose phosphate pathway. We're going to get to that. Some foods are actually made by fermentation. This is probably not news to you. But some microorganisms in foods ferment those foods, the carbohydrates in them, and they make all these metabolic products that give the food their taste and texture and all those things. And the drop in pH is what actually keeps the food from spoiling. So other microorganisms don't like to hang out in low pH. These are some examples of fermented foods and some of the microorganisms that help ferment. So lactobacillus, you see that all the time on yogurt. So the reason why is because it ferments carbohydrates in milk to make lactic acid, which makes yogurt. There's all kinds of microorganisms that are involved in making cheese. There's pickles, kimchi, kombucha, soy sauce, all these things that we enjoy, fermented foods. They're tasty. They have kind of a tang from the acid, and their textures are all different. You know, sometimes you pickle something and it gets crunchy, whereas you're making cheese, it gets kind of creamy, or you could have a harder texture cheese. So all those things are related to what microorganism is at play and all the different intermediates that are present in what amounts. So it's a whole, I mean, food science is real, and I think it's really cool. So those were the fates of pyruvate. Now we're going to talk about gluconeogenesis. Gluconeogenesis is the pathway that converts pyruvate and other 3 and 4 carbon compounds to glucose. So this is our way around the fact that glycolysis is irreversible. Gluconeogenesis occurs in all kingdoms of life, and it mainly occurs in the liver in mammals. So there are some steps that are shared between gluconeogenesis and glycolysis. And in a nutshell, the reactions in glycolysis that are reversible, those are typically the ones that you're going to see in gluconeogenesis as well. So these pathways, again, are not identical. I can't stress that enough. They're not identical pathways running in opposite directions. They're actually 2 separate pathways that share a few steps. Because some of the reactions in glycolysis are essentially irreversible, you can't use those to do gluconeogenesis. So you have to get around those by using other exergonic reactions. This figure here on the right gives you the summary of glycolysis and gluconeogenesis, and it allows you to compare the enzymes and the processes that are going on to get from glucose to pyruvate and pyruvate to glucose. Remember that you need 2 pyruvate molecules to form 1 glucose molecule. So the essentially irreversible glycolytic reactions have really big negative delta Gs. So making that glucose 6-phosphate, big old negative delta G. Making fructose 1,6-bisphosphate, remember that that was the step, that's the first committed step. Big negative delta G. And the other really big one is making pyruvate from PEP. You can't overcome those. You cannot drive those in the reverse. They don't want to. They will not. So how do we get around it? we've got to convert pyruvate to phosphoenolpyruvate. And we're going to use two reactions to do that. So the pyruvate molecules are transported from the cytosol into the mitochondria, or they're generated from alanine within the mitochondria by transaminase. We're going to talk about transamination later on when we talk about amino acid biosynthesis. Pyruvate carboxylase is a mitochondrial enzyme, and that converts pyruvate to oxaloacetate. Oxaloacetate is going to show up again when we do the citric acid cycle. So if you're like, I feel like I've heard that before, you have. So this enzyme requires the coenzyme biotin. You may have heard of biotin as a ligand for streptavidin. It's a very, very tight binding pair. So biotin is, you'll see it in like kind of hair, skin, and nail vitamins, like take your biotin, it'll make your hair stronger and shinier and all that stuff. So the overall reaction that is done here with this enzyme is you take pyruvate, bicarbonate, and ATP, and you form oxaloacetate, ADP, and inorganic phosphate. So that's the overall reaction. Now let's look at it a little bit more closely. We've got bicarbonate, and it's converted to carbon dioxide by using ATP. That's what happens in site 1. And that carbon dioxide binds to biotin. Biotin is actually attached to a lysine residue that is in the pyruvate carboxylase active site. So we've got this carbon dioxide bound, and the biotin actually swings around to deliver the carbon dioxide to the second site within the pyruvate carboxylase enzyme. So there are two sites. The first site, you have carbon dioxide binding after we made it from bicarbonate. Then we have this biotin arm that swings around and delivers the carbon dioxide to the second site. Now think about why you might need a long arm or something to hold on to carbon dioxide to do the next reaction, which is to deliver that carbon dioxide to pyruvate to elongate it and form oxaloacetate. So think about why we might need a cofactor to hang on to that carbon dioxide. We have pyruvate. Resonance is always a big deal here. So remember that pyruvate can be in the keto form or the enol form. In the enol form, you have a hydroxyl, which if it is deprotonated, it can form a double bond with the carbon to make the keto form. And in doing so, hop out, you can take these electrons from this double bond and attack the carbonyl carbon in CO2. That's an electrophile. It's got two oxygens, right? So that carbon, it's got a big old partial positive on it, ready to be attacked. And in doing so, you create oxaloacetate by adding on that CO2 to the end of the molecule. So we've made oxaloacetate. Now what do we do? Well, the mitochondrial membrane doesn't have a transporter for oxaloacetate. So we've got to do something else to it so that then we can shuttle it back out and do some more chemistry. And that something else is to use malate dehydrogenase, and that takes NADH and reduces oxaloacetate to malate. It is readily reversible. So you can go back and forth, and that's okay. If malate looks familiar, you're seeing some of the intermediates and some of the enzymes that are used in the citric acid cycle. So if you feel a little bit like deja vu, that's okay. So the overall reaction is here. We've got oxaloacetate, NADH, and a proton. We slap them together with that dehydrogenase and we make malate and NAD. So malate leaves the mitochondria through a transporter because malate can be moved about the cabin. Oxaloacetate, there was no transporter. So once it leaves the mitochondria and it is in the inner mitochondrial membrane, so that's where the transporter is, it moves through that membrane and then it gets reoxidized to malate. Sorry, y'all. Let me try that again. So malate is actually going to leave the mitochondria through a transporter that lives in the inner mitochondrial membrane. Once it leaves, it gets reoxidized to form oxaloacetate. And you also make NADH. Now that we've got oxaloacetate outside of the mitochondria, we can move on to the next step, which is converting oxaloacetate to PEP. This uses PEP carboxykinase. You need magnesium, and you also need GTP. GTP, you can think about it as just like ATP, so you can use it to transfer phosphoryl groups. And this reaction is reversible under intracellular conditions. The overall reaction is below. You've got oxaloacetate and GTP. You form PEP and carbon dioxide and GDP. So overall, to bypass the first reaction, remember that we couldn't just take pyruvate and slap them together. We had to do something else. So we're taking pyruvate, ATP, GTP, bicarbonate, and we're making PEP, ADP, GDP, and phosphate and carbon dioxide. So not exactly the greatest thing to do. We're using a lot of energy, right? But the actual free energy in the cell is negative. So that makes this reaction pretty much irreversible under cellular conditions. So if we were just trying to do this on the benchtop, it would not be fun. Okay, we'd have to give this reaction some energy and it would be reversible. But in the intracellular conditions where this is happening, mainly in the liver, it's going to happen if it needs to happen because of that spontaneity due to the negative delta G with the actual cellular conditions. So there are alternate paths from pyruvate to PEP. When lactate is the glucogenic precursor, which just means it's what's going to be used to make glucose, then there's a second bypass that predominates. Oxaloacetate is directly converted to PEP in the mitochondria by an enzyme called PEP carboxykinase. So if you look at the cycle here, we've got pyruvate in the cytosol that gets pumped into the mitochondria. We make oxaloacetate, then we have to make malate, which then gets pumped back out into the cytosol, and then you make PEP. That was what we just talked about. On the other side, if we use lactate, lactate can be used to make pyruvate, and then pyruvate goes into the mitochondria. You make oxaloacetate, and you make PEP, and then that is what's transported back into the cytosol. So this figure here on the right is a good summary of the two different paths that you can get from either pyruvate or lactate to phosphoenolpyruvate. Now we've got to talk about the other bypasses that we have to make. So fructose 1,6-bisphosphatase converts fructose 1,6-bisphosphate to fructose 6-phosphate, and it hydrolyzes the phosphate group on the C1. You need magnesium. It is irreversible. Glucose 6-phosphatase. That catalyzes the hydrolysis of glucose 6-phosphate to glucose. You need magnesium. It's only found in the lumen of the ER of hepatocytes, renal cells, and epithelial cells of the small intestine. So it's only in a few places that you're going to find this enzyme. And it's going to hydrolyze that phosphate group. Hydrolyze just means that you use water to cut. And you generate glucose and free inorganic phosphate. When you sum up all of the parts of gluconeogenesis, and we didn't by any means go through every single step of gluconeogenesis, we just went through the different reactions that are bypassed in glycolysis to make gluconeogenesis the reverse. So go back to that first slide that had the comparison of glycolysis and gluconeogenesis to see where the reversible enzymes fit into the whole scheme of gluconeogenesis. If you sum up the reactions to go from pyruvate to free blood glucose, you've got pyruvate, which is a high energy molecule, plus a ton of energy, water, protons, to make glucose. So you are expending energy to make glucose. Very, very energetically expensive. So these are the reactions in sequence starting from pyruvate for gluconeogenesis. You'll notice that the ones in red are the ones that are bypasses. The ones in black are the ones that are reversible from glycolysis. So they use the same enzyme, they're just doing it in the opposite direction. There are also some amino acids that can be used to make glucose. They're called glucogenic amino acids. You do not need to know what all of these are. Just understand the concept that there are amino acids that can undergo a conversion to glucose. The intermediates of citric acid cycle can also undergo oxidation to oxaloacetate, and then oxaloacetate can be used to make PEP. So mammals can actually convert fatty acids to glucose. We just can't. We don't have the enzymes to do it, so we can't take acetyl-CoA and make pyruvate. But plants and microorganisms have the glyoxylate pathway, and that allows them to make glucose from fatty acids. This is more for your knowledge. I'm not going to really be testing you on this. Can mammals convert fatty acids to glucose? True or false? No. This is just for your knowledge to make a complete story. We're talking about all the different ways that we can feed into gluconeogenesis, so we need to kind of be complete if we're talking about more than just mammals. Last section of chapter 14, and my favorite. Actually, I really like this chapter, so I can't say it's my favorite. But we're going to look at the regulation of glycolysis and gluconeogenesis. These two processes are the reciprocal of each other. And if you had glycolysis going in one way, gluconeogenesis in the other, if you have them both going at the same time, that's pretty wasteful. So regulation is what prevents operating both of these pathways at the same time. We absolutely need regulation here. We talked about hexokinase and how there are different isozymes. Well, those isozymes are affected differently by their product, glucose 6-phosphate. So hexokinase 1, 2, and 3 are all inhibited by the product that they make, glucose 6-phosphate. So if you have a lot of glucose 6-phosphate around, you're not going to keep making glucose 6-phosphate. You're going to pause a little bit. Hexokinase 4 is in the liver, and it's not inhibited by glucose 6-phosphate. It's its own beast. So the one in the liver, which is also called glucokinase, it has kinetic properties that are related to its larger role in maintaining glucose homeostasis. Because remember, the liver will release glucose to regulate blood sugar. So the Km is higher than the usual glucose concentration. If you look at, let's say that our Vmax is somewhere around here. So half Vmax, somewhere around there, which means our Km is somewhere in the ballpark of 10 millimolar. Remember that the Km is the substrate concentration at half Vmax. We can only guesstimate Vmax here. But that is much higher than the normal fasting blood glucose level. So there's a regulatory protein that anchors hexokinase 4 inside the nucleus. So it's kept away from all the other glycolytic enzymes. Remember that the liver still does glycolysis, just like all the other cells. So it's got to keep this hexokinase separate from everything else. What affects hexokinase 4 is fructose 6-phosphate. So let's look at this figure down here below. We can have the uptake of glucose through a transporter, and that glucose can then be phosphorylated to make glucose 6-phosphate, and then we can have the rearrangement happen, make fructose 6-phosphate, just like we would in glycolysis. But what happens is this molecule will say, hey, maybe you need to stop making glucose 6-phosphate. So we're going to tell you, maybe you should go back and hang out in the nucleus with your regulatory protein and stop making glucose 6-phosphate. If, however, there's a lot of glucose and not very much fructose 6-phosphate, then you're going to get a signal that says, hey, so dear nucleus, we need that hexokinase 4 out here so that we can make some more glucose 6-phosphate. Would you please send it out here and leave that regulatory protein behind? So fructose 6-phosphate tells it, hey, go back to the nucleus, stop making glucose 6-phosphate. And glucose tells the hexokinase 4 to come out wherever you are and start adding on a phosphate to the glucose. Now let's talk about another enzyme that is regulated heavily, phosphofructokinase 1, or PFK1. And we also have fructose 1,6-bisphosphatase. They are reciprocally regulated. So phosphofructokinase, that is in glycolysis. We are trying to make energy. If you have a lot of ATP around already, you probably don't need to keep doing glycolysis. So ATP binding actually inhibits PFK1. It binds to an allosteric site that is away from the active site of the enzyme. ADP and AMP will relieve this inhibition. So if you have one of those two molecules bound, then you'll actually activate the enzymatic activity of PFK1. Another chemical that will regulate PFK1 is citrate. So citrate is something from the citric acid cycle. And if you're doing a lot of turns of the citric acid cycle, then that means that you've got a lot of energy too. So that is also going to inactivate PFK1. Whenever PFK1 is inactive, then fructose 1,6-bisphosphatase is probably going to be active. So like I said, citrate is a key intermediate in aerobic oxidation. So we're talking about the citric acid cycle. High concentrations will ramp up the inhibitory effect of ATP. So those two signals together will stop PFK1 in its tracks. It tells the enzyme, hey, we've got enough energy, so hang on. We don't need to do this anymore. Now let's talk about the other enzyme, the phosphatase. It is inhibited by AMP. So if you have high concentrations of AMP, then that means that you're in a low energy state. So you don't have a whole lot of energy to throw around. That's going to halt glucose synthesis. So remember that these two enzymes, PFK1 and this phosphatase, they are in competing pathways. And if we have low energy, we don't want to do the high energy output of glucose synthesis via gluconeogenesis. It just doesn't make sense. However, if we've got high ATP, that's going to slow down glycolysis by slowing down PFK1, and it's going to speed up gluconeogenesis because that is going to increase the activity of the phosphatase. So you can create more glucose, and that's okay. We've got energy to spare. So if you understand the regulation of one molecule, then you'll understand why the other enzyme is the opposite, because they're in two competing pathways. Let's talk about some other allosteric regulators. We've got glucagon, which I remember all these years, glucagon, glucose is gone. So that means you've got low blood sugar. It signals to the liver, you need to make glucose and release it to the blood. Stop using it for glycolysis, liver. Share your glucose and increase the blood glucose level. Insulin is the reverse of that. It tells the liver, hey, take up all that glucose, use it as fuel, and while you're at it, you can store some of this glucose as glycogen if you'd like. You could also make some triacylglycerol. So that's in the build us up, replenish our stores type of phase. And that's released when blood glucose levels increase. So think about after you eat a meal, your insulin level increases. You're taking all of the sugars in your blood and pulling them into the cells to do glycolysis, and your liver is doing glycolysis and storing that glucose for later use as glycogen. Fructose 2,6-bisphosphate is also a regulator. It regulates both glycolysis and gluconeogenesis because it regulates PFK1 and the phosphatase. I say the phosphatase because FBPase1 is just too difficult. You heard how I struggled. A B and a P right next to each other. I can't. So I call it the phosphatase. So fructose 2,6-bisphosphate binds to PFK and it increases the affinity for fructose 6-phosphate. Increasing the affinity means that you're going to increase PFK1 activity. It does the opposite for the phosphatase. it binds and reduces the affinity for its substrate. So it decreases the activity. One molecule, two different responses, depending on which enzyme it's bound to. I'd say that's pretty efficient. Instead of having 10 different signals for 10 different enzymes, maybe you have, 4 or 5, and they all do different things depending on what they're bound to. So this is just showing in terms of the kinetics and looking at the activity and the amount of fructose 6-phosphate, what happens to glycolysis and gluconeogenesis by way of what happens through these major pathways. So remember, PFK1 catalyzes the first committed step of glycolysis. So that's a very major gateway to control. When you have fructose 2,6-bisphosphate, you can see that the activity is like crazy, okay? Whereas without it, or very, very low concentrations, you see almost no activity. The opposite is true for the phosphatase. No fructose 2,6-bisphosphate, we've got a lot of activity. I mean, that's almost straight up into the air, okay? The slope on that is pretty steep. So that's a very fast reaction. However, you add in that fructose 2,6-bisphosphate and it slows right down. So if you're a visual creature, I would say these graphs would kind of help you visualize what's going on in terms of the kinetics and when, one enzyme is turned on and one is turned off. And also remember to relate it to whether or not glycolysis is going to be the predominating pathway or if it's going to be gluconeogenesis. Since this molecule, fructose 2,6-bisphosphate, is really, really important because it tells glycolysis, hey, you've got the green light, and gluconeogenesis, you need to ease up, it needs to be regulated too. And that's where another phosphofructokinase comes in, PFK2. It catalyzes the formation of fructose 2,6-bisphosphate. And there's a phosphatase that breaks it down. Starting to see a trend here, right? There's usually one that builds up, one that breaks down, and they regulate some really crazy important process. So if you have fructose 6-phosphate, PFK2 is going to take an ATP, and it is going to phosphorylate fructose 6-phosphate to make the bisphosphate product. The phosphatase takes the bisphosphate product and will snatch off one of those inorganic phosphates to make fructose 6-phosphate. Let's say that we have a lot of fructose 6-phosphate. We're going to be stimulating glycolysis. But what does that mean in terms of looking at these enzymes, PFK2 and the phosphatase 2? You know what I'm talking about. Phosphorylation regulates a lot of different enzymes. And phosphorylation is the difference between PFK2 being active, meaning that you're making more of the fructose 2,6-bisphosphate, and FBPase, or the phosphatase 2, being active. So a difference in phosphorylation is what regulates this whole cycle. If FBPase2 is phosphorylated, it is active. That means that you're going to be degrading the fructose 2,6-bisphosphate and creating fructose 6-phosphate. That's going to stimulate gluconeogenesis, and it's going to halt glycolysis. However, if you have a phosphatase that cleaves that phosphate group from PFK2. Now you've got active PFK2 and inactive phosphatase. This reaction is stimulated by insulin because insulin wants you to take up all that glucose, do glycolysis, store the glucose as glycogen, all those things. So insulin promotes the dephosphorylation of PFK2. If we have an enzyme, a kinase, that phosphorylates the enzyme, then you've got active phosphatase and inactive kinase. That's stimulated by glucagon, which makes sense because glucagon, glucose is gone. We need to make more to put out into the bloodstream. And the way that we make more is gluconeogenesis. So glucagon activates a cyclic AMP dependent protein kinase, which increases the amount of cyclic AMP, and that activates the cyclic AMP activates the protein kinase, which then goes and phosphorylates the enzyme, and then you've got the active phosphatase. So knowing how phosphorylation affects the activity of these enzymes is key. So we talked about glucagon and insulin on the other slide with the figure. This is it in word form. So if you like to read words or you like to highlight words, here are the words for you. So I'm not going to go through and reiterate what we just talked about on the last slide. Another key regulator of metabolism is xylulose 5-phosphate. It's an intermediate of the Pentose phosphate pathway, which we're going to talk about, and that activates phosphoprotein phosphatase 2A. What this enzyme does is it dephosphorylates the PFK2 phosphatase enzyme. This causes an increase in the concentration of fructose 2,6-bisphosphate. Remember, if you have an increase in this molecule, you're going to increase glycolysis. The glycolytic enzyme pyruvate kinase is also allosterically inhibited by ATP, and that makes sense. If you have a lot of ATP, you don't really need to go through the glycolytic pathway to make more energy. And you certainly don't need more pyruvate to go through the citric acid cycle and oxidative phosphorylation to make, again, more energy. So in general, when you think about the overall metabolic control, ATP tells the cell, high levels of ATP tells the cell, hey, we're good on energy. Stop all of these glycolytic enzymes. Instead, turn on gluconeogenesis so that we can make some glucose and we can store it and do other things. In the liver, remember the liver is special, glucagon will activate protein kinase A. And then that will again phosphorylate pyruvate kinase, and it is inactive. If it gets dephosphorylated, then it becomes active. That's only in the liver. So this is one of the ways that pyruvate kinase is regulated. Phosphorylation puts a hold on it. Take that phosphate off, we're ready to roll. In all the other glycolytic tissues, so everything else that makes pyruvate using glycolysis, including the liver, we've got ATP, acetyl-CoA, and long-chain fatty acids all regulate pyruvate kinase. They all say, hey, you don't need to do this anymore. We've got a lot of acetyl-CoA. We're making a lot of stuff. we don't need to make more energy. We're good. However, fructose 1,6-bisphosphate says, hey, you should turn on. So the pyruvate kinase will be turned on in the presence of fructose 1,6-bisphosphate because it says that glycolysis is active and we need to continue on through and make pyruvate. All of this regulation is something that you're going to have to sit down with and kind of, it's helpful to make some kind of a chart or some kind of a figure that shows how all of these hormones regulate the different enzymes and what that overall hormonal response means, whether you're turning on glycolysis or turning on gluconeogenesis or what have you. So kind of sitting down with all of these little figures and trying to synthesize it all into one humongo figure that talks about the ways that insulin affects all the different enzymes to turn them on and how glucagon affects enzymes to turn them on or off, etc. So conversion of pyruvate to PEP is stimulated when fatty acids are available. Acetyl-CoA says, hey, we don't need to make any more pyruvate. Glucose oxidation, we don't need you. We don't need your glycolysis because we've already got a lot of acetyl-CoA to start doing our anabolic processes. So it actually allosterically stimulates pyruvate carboxylase to make oxaloacetate and begin gluconeogenesis. It also inhibits pyruvate dehydrogenase, which means that you're not going to take pyruvate and make acetyl-CoA, which can then go into the citric acid cycle, because we said, hey, we've got enough of this. So transcriptional regulation changes a lot of enzyme concentrations. I don't need you to know all of the things that are in this table, but just understand that insulin regulates a lot of genes to turn on glucose metabolism. So just make sure that it makes sense to you why insulin would turn on genes that express proteins involved in glycolysis and why it may turn off the genes that are essential for gluconeogenesis. We've also got CREB-P, carbohydrate response element binding protein. It's a transcription factor that's expressed in the liver, adipose tissue, and the kidney. And it has to act in the nucleus because it's a transcription factor. That's where they kind of have to go. That's where your chromosomes are. And it regulates the gene expression for genes encoding the enzymes needed for carbohydrate and fat synthesis. So it's a pretty major thing. When you have glucose come into the cell via transporter or some other mechanism, it's automatically going to get, and this is in the liver, it's going to get phosphorylated, so it's trapped in the cell. You can go through the pentose phosphate pathway and make xylulose 5-phosphate. That's one of the intermediates, which will turn on a phosphatase that removes these phosphate groups on the binding element. Once those phosphates are removed, actually it only removes one, not both. Got a little bit excited there with crossing things out. So once you remove, and I should probably make the correct one. This isn't exactly to scale, but I want to try to stay consistent. There we go. Once you remove one of those phosphates, now this transcription factor can be translocated to the nucleus where it can bind DNA along with some other factors after it gets completely dephosphorylated, and it can increase the gene expression of all the things for glycolysis and fatty acid synthesis. So it's going to bind to transcription factor binding site and make that DNA more accessible for the machinery to create that messenger RNA transcript. You don't need to know about how the increase in all the gene expression actually happens, because we haven't covered that yet. But you do need to understand what happens to get this Krebs protein into the nucleus to allow it to bind to its binding spot on the chromosomes. So that's it. This is part 2 of chapter 14. There's still one more part. We have to talk about the pentose phosphate pathway. The regulation is going to be a key thing where you're going to really have to sit down with all of this information and put it together so that you understand how all the pieces work together. The body is very complex, and there are so many different processes that have to be regulated, so it's sometimes hard to wrap your mind around. But if you take it slow and really try to recreate some figures, try to synthesize information into kind of a more compact form, so you don't have to find 8 different figures to figure out how insulin affects all of the enzymes in glycolysis. So really do your work there to put all this information together. It'll help you a lot more when you're studying and doing questions for assignments. So with that, thanks again for watching, and be safe.